Okay, so thank you everybody for attending the Phi Tau Sigma February webinar of the 2025-2026 webinar series. Um, so I just wanted to have some sort of announcements. So just as a heads up to mark your calendars for Friday, March 27th, uh, almost a month from now, we'll be having Dr. Brendan Namira, who's actually the Chief Science and Technology Officer of the Institute of Food Technologists, giving a talk about some of the awesome decades of research that he's done on non-thermal food processing and control, as in particular cold plasma. So that should be a really, really interesting webinar. Just as a heads up for folks, these webinars are available on YouTube at the address listed below. And it's without further ado that I'm really honored and excited to get to introduce Dr. Jianan Zhang, who will be giving our webinar today, entitled Molecular Insights into How Diet and Xenobiotics Impact Gut Health. Dr. Jianan Zhang is an assistant professor at the Department of Food Science and Technology at University of California, Davis. Um, Dr. Zhang did her BS in Food Safety and Quality as well as a BA in English at Huaqiang Agricultural University in Wuhan, China. She then did a Master's of Engineering at China Agricultural University under Dr. Baoping Ji. And she did her PhD here at UMass in the Department of Food Science under Dr. Gudong Zhang, where I was privileged to get to meet her, and she was a star in our department and did lots of really high-powered research. Dr. Zhang then went on to do serve as a postdoctoral research associate in the Department of Chemistry at University of North Carolina, Chapel Hill under Dr. Matt Redenbaugh. She's won many, many awards. She has 29 publications, including in super high-powered journals like Nature Communications. She's won numerous awards associated with her research as well as her stellar academic record, including the 2021 Phi Tau Sigma Founders Award. So it is a very, very decorated research career. She has 4 patents and is actually serving currently as an associate editor for Frontiers in Nutrition, as well as a guest editor of a number of really interesting special issues there. And so it's really my honor and privilege to get to introduce Gina. And I'm really excited to hear about the really awesome work that she does. So I'll stop sharing my screen and feel free to take it away whenever you'd like, Gina.
Thank you so much for Dr. Moore's kind introduction. Yeah, it's really exciting for me to share all my research with everyone here. And I'm Jianan Zhang, and I'm recently just hired as a assistant professor at the University of North of California, Davis, in the Department of Food Science and Technology. And today I will walk you through my research during my PhD and postdoc. And my talk today is focusing on how molecular insights into how diet and xenobiotics impact gut health. So we all heard about that an apple a day keeps the doctor away. And this is also the inspiration for me to dive into the food science like many years ago. And as we all know that apples are rich in fiber, polyphenols, antioxidants, all these beneficial compounds for human health. But after many years of study, I put a question mark for these sentences. An apple a day really keep the doctor away. On the other hand, the apple is also rich in fructose, sorbitol, which are actually can causing bloating, discomfort, and diarrhea to people who have certain gut health issues. For example, for patients who have irritable bowel syndrome, short for IBS, or inflammatory bowel disease, short for IBD. And these patients are actually, they should avoid eating apples during their flare. So this comes to say that one size fits all does not work, particularly for those with gut health conditions. So gut health is actually being influenced by many factors. And in fact, there are 1 out of 10 adults in the United States have a certain type of gut health issues. For example, the genetics, the stress, the quality of sleep, the lifestyle, smoking, probiotics are all the factors that can influence the gut health. And of course, diet. And also the usage of antimicrobial additives and medications can also influence gut health. And as we all know that we are not alone. Gut health is not only about host metabolism, but also we need to understand microbiome metabolism. And as a matter of fact, that there are over 10 trillion microbes living together with us for every second. So for today's talk, we want to understand how gut health being influenced by three major factors. First one is the diet, and second one is antimicrobial additives, and the third one is medication. So today I will walk you through about these three major factors that influencing gut health. So let's start it with the how diet is influencing colon cancer. Colon cancer is ranking the third of all different type of cancers, both men and women, as you can see from this table. And what makes it even worse that colon cancer is actually increasing in younger adults, which are younger than 50 years old, both men and women. And linoleic acid is actually an essential fatty acid to our life, and it is the most abundant polyunsaturated fats in the Western diet. And they are rich in all the seed oils, for example, the corn oil, soybean oil, and sunflower oil. And the consumption of linoleic acid is actually increasing for the past 100 years. So they started from 3% of calories of linoleic acid into around 9% of calories. These are due to the highly development of the oil extraction and oil processing. However, and the consumption of linoleic acid is actually high globally, as you can see from this map, and especially from the deep green areas, they are over 10% of calories. However, the high consumption of linoleic acid is actually a health concern. A very recent publication from the Cornell Medicine School, they noticed that high consumption of linoleic acid is actually a concern for breast cancer, showing here using in vivo model, and they found that it is linked to the fatty acid binding protein 5 pathway. And so the big question for the first story is, does high consumption of linoleic acid promote the development of colon cancer? To run that, we designed two types of the diet. One diet is control diet, which is rich of 3% of linoleic acid. And the second diet is linoleic acid rich diet, which contains 9% of linoleic acid to better mimic the consumption of human as well. We run this study in a mice model, and we are using a AOM-DSS-induced colon cancer model. AOM-DSS is a widely used reagent to inducing colon cancer in mice. So we treated the mice with these two different diets for 12 weeks. And after 12 weeks, we collected the colon tissue and their plasma and to run the analysis. And here is what we found. This is the colon tissue, and we noticed that compared to the control diet, the linoleic acid-rich diet have the significant higher tumor number in the colon and also the tumor size. So we say linoleic acid-rich diet can exacerbate the development of colon cancer. And additionally, we also run another study using healthy mice, or we say it's a basal mice model, and treated with these two different type of diet, and we found minimal toxicity. So again, here we show that we need to separate the disease model when we treat diet. We need to consider whether they are healthy or they are vulnerable population, such as the colon cancer patients. So our next question is, which fatty acid metabolites are affected by the linoleic acid-rich diet? And linoleic acid is actually one of the polyunsaturated fatty acid. They can be further metabolized into different lipid mediators via three major enzymatic pathways. First one is the COX pathway, second one is the LOX pathway, and the third one is the cytochrome P450 and the soluble epoxide hydrolyse pathway. And they can convert it into various lipid mediators, which has profound effects in human health. The way we detected the metabolism of the fatty acid is that we are using LC mass-based lipidomic to detect the metabolism. For this model specifically, we collected the colon tissue and also the plasma from the two different type of the diet-fed mice group, and then we run the LC mass-based lipidomics, and then we detected the lipid metabolites. And in this model, we actually successfully detected over 60 of the metabolites. And around 60 metabolites in plasma and colon, we found 16 of them were significantly changed by this linoleic acid-rich diet, showing here in these two figures. And the left one is the plasma, and the right one is the colon. And among all the 16 metabolites that were altered by this linoleic acid-rich diet, we noticed that the most dramatic changed metabolites derived from the linoleic acid is actually the 12-13 EPOM and 9-10 EPOM. So what is the EPOM? EPOM is actually the metabolites derived from linoleic acid via the CYP pathway. So we can say that linoleic acid-rich diet can promote the development of colon cancer. And we found that the linoleic acid-derived metabolites, EPOM, were the most dramatic increased metabolites. So to better understand, the CYP enzyme is a key enzyme for linoleic acid converted to EPOM. We further analysis, we further ask the next question is, what would happen if we block one of the enzymes, which is specifically for linoleic acid, is the CYP2C or CYP2J, and using a genetic modified mice model, what will happen? To run that, we collaborated with Dr. Darryl Zeldin at NIHS, and we have the CYP2C heterozygous mice. At the same time, we have the wild-type mice. And we run the exactly same mice model. We fed these two different type of mice with control diet or linoleic acid-rich diet and treated with a AOM DSS-induced colon cancer model and fed them for 12 weeks. And after that, we collect the tissue and run the analysis. And here is the results. We found that in the wild-type mice, compared to the control diet, linoleic acid can significantly increase the tumor size, which is consistent to what we found in the previous model. At the same time, we noticed that the CYP2C heterozygous mice show that the linoleic acid diet can alleviate the tumor size in the colon. So we say this CYP monooxygenase is required for the colon cancer promoting effects of this linoleic acid-rich diet. And this is still not the end of the story, and because EPOM can be further metabolized into dihome via the soluble epoxide hydrolyse pathway, and all these compounds are very active and unstable. And our next question will be, is it the EPOM that derived from linoleic acid or the dihome that derived from the linoleic acid that is causing the colon cancer promoting effects in the mice? And because, as I just mentioned, that EPOM and dihome, these lipid mediators are very active and unstable, and we are using an osmotic pump model to induce the mice, which is helping us to deliver a controlled dose of the active drug compounds, which is also used in clinical. So here is the mini pump we are using, and we have three different groups. One is the EPOM, second one is dihome, the third one is the vehicle. And then I did a little surgery at the back of the mouse and put this little mini pump at the back of the mouse to deliver these drugs. And we further run a AOM DSS model, and this time we're using a chow diet instead of the linoleic acid rich diet. And after 9 weeks of this study, we found that in the colon tissue, as you can see here very clearly, in vehicle group, the tumor number of the mice is being significantly increased in the EPOM group compared to the vehicle, showing here in red and also in gray. While treated with dihome group, showing here in blue, could show no difference compared to the vehicle group. So for this model, we showed it again and confirmed it. It is the EPOM, but not the dihome that significantly promote the colon cancer in mice. So a small summary for the first story is we found that high consumption of linoleic acid can promote the development of colon cancer via the CYP pathway, and its metabolite EPOM contributes to the pro-cancer effects. So this is the story I showed that how diet or food is influencing gut health, mostly via the host metabolism. And my second part of the story will show how xenobiotics, specifically how antimicrobial additive, been influencing gut health via the microbial metabolism. So triclosan is one of the most used antimicrobial additive to in our environment or in our life. This is a most used antimicrobial additive in the toothpaste, in the hand soap, or in the cosmetics, and even in children's toy. And also, it is a widely used preservative. And let's make it short, is triclosan is broadly released into our environment. And however, triclosan is a risk factor for colon inflammation and colon cancer, which been found by our group. And here it shows that when we exposure the mice with triclosan, it can shorten the colon length in the colitis model. The length of the colon length is a biomarker for colitis. And also, we found that the triclosan exposure in mice can increasing the tumor number in the colon and in the colon cancer model. So triclosan is a risk factor for colon inflammation and colon cancer. And we further found that the gut bacteria are actually required for this colonic toxicity of triclosan. This is the model we use to show that in a germ-free mice, we noticed that the colonic toxicity of triclosan is alleviated, while in the conventional mice, you can see that the triclosan can causing the damage of the colon tissue in this histological analysis. So we say the gut bacteria is required in this colonic toxicity caused by the triclosan. But how gut bacteria contribute to triclosan-induced colonic toxicity is not been defined. So the big question of the second story is that what is the role of gut bacteria in triclosan-induced colonic toxicity? Before we dive into the data part, we can share about the metabolism of triclosan in vivo. When triclosan get into our body, they actually will convert it into conjugated form of triclosan into more hydrophilic form, mostly by the host enzymes in the liver. And these conjugated form of triclosans are actually biologically inactive. They can change into, for example, the triclosan glucuronide, the triclosan sulfate. And so our big question for here will be how this conjugated triclosan reach to the colon and causing colonic toxicity, which is related to gut bacteria, is unclear. So our first question is that we need to identify which conjugated triclosan it is when it's reaching to the colon. So to run that, we did our first animal study. We have the mice and we let the mice exposure to triclosan for 4 weeks, and then we collected their organs and run the triclosan metabolism. And here is what we found. We actually collected the heart, the liver, the bile, the small intestine, the cecum, and the colon from both mucosa and also the digester of this GI tract. And we detected that, as you can see here, we noticed that in the cecum and the colon, there is a unique pattern that the major metabolites being found in cecum and colon is the free-form triclosan, showing here in yellow. Well, one second. Well, the major form of the small intestine, as showing here, is the blue part, which is the triclosan glucuronide. To confirm what we found, we run a human study. We have control group and also the group that are being exposed to triclosan. And we collected the urine and the stool samples from the human, from the group, and then we detected the triclosan metabolism. And here we found that in the total triclosan level in the stool samples was significantly increased in the triclosan group. This is not a surprise for sure. And what we found later is that we found that the triclosan in the stool samples, the major metabolism is the free-form triclosan, showing here in yellow. And the major metabolites of triclosan in urine is the triclosan glucuronide, showing here in blue. Hopefully you still remember what we found in the mice study, and they are consistent. We found that in the colon or the stool samples, the major metabolites of the triclosan is the free-form triclosan. So we confirm it that, again, when the triclosan reach to the small intestine, the major metabolite is the triclosan glucuronide. When it's reaching to the colon, the major part of it is the free-form triclosan. So our next question is, what is the role of gut bacteria in converting triclosan glucuronide to triclosan? To run that, we actually run a serious animal model and also the in vitro assays. For example, we use the antibiotic cocktail, treated the mice model, and also the germ-free mice model to confirm that the converting of triclosan glucuronide into triclosan required gut bacteria. And also we're using the in vitro assay confirmed that the gut bacteria is required for this reaction. And also we are even using the antibiotic cocktail treated mice model in a time course study. We found that this reaction can happen within 4 hours. So our next question will be, what is the functional role of the gut bacteria in converting triclosan glucuronide into triclosan? In other ways, which microbial enzyme is contributing to the converting triclosan glucuronide into triclosan? Of course, our first guess will be microbial beta-glucuronidase, short for GAS. And actually, the GAS enzyme, especially the microbial GAS, is very complicated. And so we run again a series of assays to figure out which microbial enzyme, which GAS it is to contribute to this reaction. I can share one of the assays we used here. We actually using the microbial proteomics to confirm or to figure out the specific gut microbial GAS enzyme. To run that, we have the human stool samples, and we run the fecal lysis and extracted the GAS enzyme and run the LC-mass-based proteomics. And this is the SMELLY study, as you can figure out. And here is the result we found. We found the triclosan glucuronide turnover rate is actually positive relative to the total gas level. And also specifically, we figure out that it is the loop 1 gas that are positive related to the triclosan glucuronide turnover rate. To let everyone have a better idea about what loop 1 gas is, I will show you a structure later. So as I just mentioned that we continued to confirm this microbial loop 1 gas enzyme is the one that converting triclosan glucuronide to triclosan. We run other assays, for example, the in vitro assay and the microbial proteomic assays, which I just mentioned, and also the crystallography assay to solve this crystal structure. And we figure out that it is the microbial loop 1 gas enzyme that converting triclosan glucuronide to triclosan. And to let everyone have a better idea of the loop 1 gas enzyme, I prepare a little movie here. So this is the microbial loop 1 gas that from E. coli. And this little loop in red in the middle, as you can see, we name it as loop 1. As you can see that it is just next to the active site, which make it a perfectly fit for the small molecular like triclosan glucuronide to make this reaction happen. So our last question for this story is that what would happen if we inhibit this microbial loop 1 gas enzyme, and will it be toxic or be alleviated to the colonic toxicity caused by triclosan? To run that, our group have identified a selective inhibitor of this microbial loop 1 gas, which we name it as gas inhibitor 9. And this is very easy to understand. It is inhibit the function of the loop 1 gas enzyme. So to run this study, we run another animal study. We used the DSS-induced colitis mice model, and we treated the mice with or without triclosan. And then we studied a DSS model to induce colitis. At the same time, we autogavaged the mice with gas inhibitor or without gas inhibitor. And after 30 days of this study, we collected the colon tissue and run the data analysis. And here is what we found. In the triglyceride alone group, as you can see here in the red, can shorten the colon length compared to the vehicle group. While treated together with gas inhibitor, triglyceride group can actually alleviate the colon length shorten in this mice model. We further run more bioactive assays to confirm what we found. Here is the flow cytometry. We are trying to detect the immune cells. Here we found that triglyceride alone group, showing here in red, can significantly increase the total immune cells, the macrophage, the neutrophil, compared to the vehicle group, while treated together with gas inhibitor can alleviate the increase of the immune cells in the colon. And we run more like gene expression to understand the gas inhibitor is working here. We found that in the TNF alpha, MCP1, interleukin 6, interleukin 17, which are all pro-inflammatory cytokines. And we found that triglyceride alone could significantly increase these markers in the colon, while treated together with gas inhibitor could alleviate it. And we also run the pathology of the colon, and we found the same results, that the triglyceride alone could damage the tissue, while treated together with gas inhibitor could alleviate it. So we say microbial gas inhibitor can protect the triglyceride promoted colitis. So a summary for the second story here will be, we identified this specific microbial loop 1 gas in reactivating the triglyceride in the colon. And we also noted that targeted inhibition of this microbial enzyme can protect against triglyceride-promoted colitis. And the very last story I want to share is that how medication is being involved in gut health via microbial metabolism. The gut microbial gas enzyme can not only activate the triglyceride, but also can work on drugs. For example, they can convert its drug glucuronide into a free-form drug and causing the GI toxicity. And one of the most consumed over-the-counter drugs, which is the such as the aspirin, ibuprofen, indomethacin. These are also named as non-steroidal anti-inflammatory drugs, short for NSAIDs. And gas enzymes can also working on these NSAIDs by converting NSAID glucuronide into free-form NSAIDs, and then causing the GI toxicity in vivo. So this is a quite serious problem, and for people who have the gut health issues, actually they should avoid using the NSAIDs. But because they are the over-the-counter drugs, and the men and women are actually consuming them in the same way. So this is quite ridiculous, right? But this is the fact. And so the sex-specific factor in the NSAID-induced GI toxicity and how the microbial metabolism being involved remained unstudied. So for this story, we are focusing on understand the sex-specific factors in these medication-induced GI toxicity. So to run that, we have two group of mice, of course, male mice and female mice, and we choose the indomethacin as the drug we are using for this study, and we treated the mice with the indomethacin, and we collected their tissues, for example, the cecum and the plasma, and we run the LC-mass to understand the indomethacin metabolism in vivo. And here is what we found, just as a small remains of this reaction that indomethacin glucuronide can be converted into indomethacin via the microbial Gas enzyme. And we found that the indomethacin glucuronate level in the plasma are actually significantly high in the females compared to the male mice. Actually, it's 10 times higher than the male mice. And at the same time, we noticed that the free-form indomethacin is actually significantly high in the male mice compared to the female mice in the sequence. So we did find they are different. The male and females have a diverse indomethacin metabolism in vivo. So what is the toxicity being caused by this drug? We further run the biomarkers for the GI toxicity, and here is the colon tissue. We found that in the male mice, the indomethacin could increase the ulcer numbers in the colon, while in the female mice, we noticed more liver toxicity or hepatic toxicity being caused in the female mice, showing here as a PPAR alpha, which is a biomarker for beta oxidation. And we found that in the female mice, indomethacin treated group can significantly decrease the level of PPAR alpha, while there's no difference in the male mice. To further better understand this, there is the sex differences between the mice when treated with the indomethacin. We also ran another Gas inhibitor study. We treated the male and the female mice with indomethacin or vehicle, and at the same time, we treated them with Gas inhibitor or without Gas inhibitor, and we collected their fecal samples to run the metagenomics to better understand the composition of gut bacteria. And here is the beta diversity we found among the groups. As you can see that there is a significant separating between males and females, and beta diversity is a marker to show the difference or the similarity between groups. As from this figure, you can figure out that very clearly there is sex-specific difference between the groups. And let's get a little closer look at this data and among all the groups. As you can see that all these male groups are all clustered together, which showing at the left part, and while the female groups are shifted. And specifically, you can see that the female group treated with indomethacin, showing here in red, and compare it to indomethacin together with Gas inhibitor, showing here in blue, shifted a lot from this figure. So again, we are showing that the sex factor is a major factor here that we definitely cannot ignore. And we also do the analysis for the gut microbiota composition here, and we noticed that even at the very beginning of this study, showing here that in the control group, males and females are already showed significant difference among their gut microbiota. As you can see that the Firmicutes level, which is the showing here in gray, and also the bacteroides showing here in blue, were different between males and female, even without treated any indomethacin or Gas inhibitor. And we further, of course, ran all the data analysis for the groups treated with indomethacin and the Gas inhibitor. And let's get a little closer look at these data. And again, as I mentioned, in Firmicutes and bacteroid data, we show that significant difference between male and female, no matter they treated with Gas inhibitor or without, or with indomethacin or without. And what we surprisingly, we found one that is significantly changed is the verrucal microbiota. We noticed that these actually were altered by the Gas inhibitor only in female mice. So a small summary for this, the third story here, that the males and the females exhibited the sex-specific response to indomethacin-induced GI toxicity. I hope I convinced you that. But why, right? We still want to understand why does that happen. And until we figure out this last piece of puzzle, we found that the indomethacin is just quite like the triclosan. When it get into the in our body, they're gonna convert it into conjugated form, indomethacin glucuronide, and in the liver. And after that, they have two way out. One way is via the bile to the intestine. The second way is via the bloodstream to the circulation. And both of them are actually being regulated by estrogen. And specifically the MRP2, which is the regulator, which is downregulated by the estrogen, while the MRP3 are upregulated by the estrogen. So this could be one of the possible explanation for why this happened. And definitely more study need to be work on it, and we need to figure out why estrogen is involved with the drug metabolism. And this is definitely one of the future direction of my lab. And until now, I have shared with you that how the molecular insights into how diet and xenobiotic can impact gut health via both host metabolism and microbiome metabolism. And I have briefly talked about the how diet being influencing colon cancer, and how triclosan being influencing colon inflammation, and how indomethacin or NSAIDs is influencing the GI toxicity via host metabolism and microbial metabolism. And what is the insight of our story? The first one I thought is that one size fits all does not work, especially when it comes to the vulnerable populations with certain type of disease, especially for gut health. And these people definitely need specific tailored interventions. And the second one is the microbial metabolism is essential to truly understand the effect of diet and xenobiotics on human health, and also, of course, on gut health. And we not need to understand which microbiome or which gut microbiome is involved, but also the functional role of the microbe need to be further understood. And the third one is that the sex-specific factor cannot be ignored. And it is so surprising for me that this, when I go to the literature, that there's the data for the female mice or the women are so rare. So we definitely need to fix this. And we are all unique. It's just like compare apples to oranges. There's no way we should treat men and women the same way in drug or in the nutrition. So this is also a photo I took from my home, that the Tylenol. As you can see that they are designed for adults, but we need to define adults, right? We can do better. And my last part will be sharing a little bit about my vision for my lab. Our lab is focusing on food in human health and food toxicology. And our mission is to understand the role of diet-host-microbe interactions in human health. Specifically, we will target it on the phytochemicals, the seed oils, the xenobiotics such as the over-the-counter drugs and the food additives, to using the multi-omic techniques to understand the molecular mechanism behind. And we will focusing on both host metabolism and microbial metabolism for both men and women. And our unique program will definitely advance the in personalized nutrition and medication. And as just mention that we are a brand new lab, and my current stage is working on establishing my lab. And I just mentioned to Dr. Moore that I just successfully recruit my first PhD students, luckily, and we definitely look forward to any collaborations who share the same interests. And of course, there's a lot of people to thank. And first, I would love to express my just a lot. Yeah, a lot to thanks. Like, for example, my advisor, Dr. Godong Zhang, which is who is my PhD advisor at UMass Amherst, and also my great advisor, Dr. Matthew Reddingbow at UNC, who is the advisor for during my postdoc training at UNC. And also, of course, thanks to all my lab mates from both UMass and UNC. And of course, thanks to all my collaborators. Many thanks to Dr. Derry Zalden at NIHS, and thanks Dr. Eric Decker from UMass, Dr. Moore from UMass, and Dr. Shell from UMass, Dr. Lisa Minter from UMass. And also many thanks to Professor Bruce Hammock and Dr. Jun Yang from UC Davis. And thanks for our fundings. And since I still have a little time, I also want to share my third picture here that our beloved Dr. Bruce Hammock was passed away last month. And he was a giant, and it's a big loss for us, for the field, for science, for the whole world. And his contribution in lipidomics, in toxicology, is huge, and he's a legend. And we truly miss her and miss him. And also this, I really want to share one piece of memory I have with Dr. Hammock. That this is the photo I took when I was a PhD student at UMass Amherst during a conference. And Dr. Hammock was asking me, have you ever thought about to become a professor in the United States? And at that time I was, definitely not even think about that question. And I think he's the most supportive scientist and human being. And looking back, I figure out that is the seed he put in my heart a long time ago. And I really want to in memory of Dr. Hammock. And I'm happy to take any questions. Thank you all for your time.
Awesome. Thank you for the amazing presentation, Jianan. And certainly, I imagine your lab will be carrying on Dr. Hammock's work. That was a very touching tribute there. Everything I've heard about him too is exactly the same. He was a major loss. It was sad to hear that he passed away. For all the folks in the audience who are interested, please post any questions you have in the Q&A. I see we already have a really interesting question or a great question from Bin Chen. So Bin was asking in the context of your Tricoslan sort of portion of the story that you were telling, I think as a question about sort of the human study, I think Ben was sort of asking more about so you've gotten human stool. Was this part of a larger sort of human study? And could you potentially elaborate on what all you did for the humans that were treated with the Tricoslan that you sort of isolated those GUS enzymes from for that data? And Ben, feel free if I'm not interpreting the question correctly to jump in there. It's a great question though. And I have a follow-up too after that.
Okay, it's the triclosan study, right?
Yeah,
Exposure with the
I think when you're looking at the conversion of TCSG to TCS.
Yes, we actually run a human study, and we actually using the the toothpaste that consume the triclosan to do the exposure in human study, and also the toothpaste that without triclosan, and which is the control group for the human study. And we collected their stool sample and urine to detect the triclosan metabolism. And after study, we checked all the toothpaste we used at home, and yeah, throw away with the ones that within the like using the triclosan.
That's crazy. So did you dose extra triclosan into the toothpaste, or was it like just toothpaste that already had it in it that you gave them? Because like I think of toothpaste, like you only use a little bit. Like that's crazy that you got those
Yeah,
results
yeah.
out of that.
We just using the toothpaste. We did not like adding more. So just to better mimic what is exposed to us.
Wow.
Yeah,
And they
because
were just using like a normal amount of toothpaste. Like they weren't like taking the tube and just like going like
no.
Slurpee machine into it. That's crazy. Just one small little strip of toothpaste. That's
Yeah,
wild.
yeah, for about like 4 months, and then so yeah, we can detect it.
Wow, that was awesome. So Ben, I hope I got your question correctly. That was a really good one. Sweet. It seems like we did.
Oh, thank you.
Thank you again for the question, Ben. We have another question here from an anonymous attendee.
Yeah.
I'm a little bit, I apologize. I'm not really sure what this is referring to. It says, is the LA hyperconsumption itself, or could be the oxidation products from LA?
Oh, that is a very good question. Yeah, we are actually using the corn oil directly
Hmm.
purchased
Oh,
from the supermarket, and to mimic the consumption of the linoleic acid. And we detected the linoleic acid level before the study happened using the GC mass to have the fatty acid profile to adjust the amount of linoleic acid in these two diets. And could that be an oxidation product of linoleic acid? That is a very, very good question. And this is actually one part of my research, and is working together with Dr. Decker to understand the mildly oxidation of linoleic acid in the gut health using animal models. Because as I just mentioned, we are just using the corn oil we purchased from the supermarket. It is the best way to mimic for human consumption. And for the oxidation study, we actually cooked it a little bit to trying to get the mild oxidation from the linoleic acid. So hopefully I answer your question.
yeah, that's an interesting question. So the idea, so I have a question. So like, let's say I buy corn oil and then I'm using it. How long would you like after opening it and using it and how long do you think that would normally elapse for somebody who's normally using it before they should start thinking about throwing it away and getting more oil if they're
That
worried about their
that
health? I know that's a broad question,
Yeah,
but
yeah, that's a good question actually. And when I'm working on the oxidation of the specific fatty acid, is the oxidation really happens so quickly. When
mm,
I'm using the fish oil, for example, the omega-3 fatty acid, they actually can be oxidized within several minutes.
mm,
And but for corn oil, they are much better, and as they are like omega-6 fatty acid mostly. And in our study, we are, I think the major metabolite is still the EPOM. We also, because we detect all the linoleic acid derived metabolism using the lipidomics. So we did find the most dramatic change. The one is the EPOM, but not the like oxidized like derived metabolism. So for the corn oil, personally, I'm thinking, yeah, the oxygen level is the key part.
mhm.
So after I purchasing the corn oil, well, after this study, I actually do not consume any corn oil anymore. I change it into olive oil. But for your question, Dr. Moore, so my way of like think that it will be probably we need to put the I coat the big bottle of corn oil into small bottles and then to minimum the oxygen level in the bottle is a probably is a practical way to protect the oxidation.
Yeah, well, so I'm not like obviously I'm not like nutrition is not my background or anything, but like all you hear about is like, oh, these are the good fats, these are the good oils to consume. Given what you've sort of revealed with like oxidation and stuff, should we just go back to eating saturated fats, or are they still just like there's still too many? I know it seems like a balance because there's like a gray area. I don't
Yeah.
know. What's your sort of opinion on that? I don't want to like get you in trouble with like the corn industry or anything, but
Yeah.
but just I'm
That's
just curious, like, well, how do you balance that now? Because now we're finding it seems like a lot of your talk, the theme was like, oh, well, we thought these were good, or we thought this all applied to one group of people, but it looks like it's maybe more of a gray area. And it seems like that's what a lot of your research is revealing is that as you dig deeper, it seems like things are a little bit more complex than just these are good, these are bad.
Yeah, yeah, definitely. I feel actually the same way. And I think from my study, at least we can concluded that for the people who have gut health issues should decreasing the level of linoleic acid. As I also mentioned, we also run a healthy mice model, which is to mimic the average of the human consumption, and we found that in the healthy mice model we did not see like the toxicity caused by linoleic acid with the high consumption. Well, we do see that this is the toxicity being caused for the people who have the or the mice who have the colon cancer, and definitely for this vulnerable population they probably we need to like tailor the intervention of their nutrients and also their guidelines for the diet of food. And then, of course, I will continue to work on the saturated fatty acid and olive oil. So hopefully I can like answer your question better, like probably several years later. Yeah, I will definitely continue working on this edible fat and oil and get a better answer for that.
Cool. Awesome. Well, it's great work. I look forward to it. I guess if anybody else in the audience, last call for questions. I see we're running up on the hour.
Oh
If there
yeah.
are no other questions, it was a great talk. Thank you so much, Dr. Zhang, for being willing to give us a talk. And again, folks, I will be posting this on YouTube if you want to revisit anything or share the link with your friends if you found it interesting. And so I just like to, on behalf of everybody in Fight House Signal, like to thank you again for giving such a great talk and taking the time to show us some of your really cool research. Really excited for the future of what's to come. Thank you very much.
Thank you. Thank you very much, Dr. Moore.
Awesome. So I'll go ahead and stop recording now.
Okay,
Um,
awesome.
let's see.